Good morning, everyone. We'll start now our last session. Just a quick announcement. Before we go to the coffee break, I will ask everyone to, when we get out of here, we'll take a picture of the group by the dome over there. So we'll start now our section. So.
Okay. Good morning to everybody. Let's start the morning section. It's a pleasure for me to introduce you Dr. Rafael Bernardi, working in computational biophysics approaches to mechanosensing. He is from the Department of Physics at Auburn University in Alabama, USA. Thank you, Rafael. The audience is yours now.
Okay. Thank you. You can hear me, right? So first, thank you for the invitation, for the organizers, for inviting me for presenting here. There is an interesting fact that I mentioned to some people that my first ever conference or school of physics in my life was actually in this place. It was not this building, it was the older IFT building, and that was about 20 years ago, so it was kind of interesting to come back here. I'm gonna show to you a little bit about what we are doing on computational biophysics for on using molecular dynamics and some analysis of molecular dynamics in a different way for studying mechanoproteins. So when we talk about studying mechanoproteins, typically you're going to think about some single molecule methods like single molecule force spectroscopy, and that is done by using, you can use an AFM, an optical tweezer, like magnetic tweezers. You can use centrifugal force microscopy. That is kind of a new one. And there's a bunch of different other methods that you can use to do it. If you're using an AFM, this methodology was created by Gaub in the 1990s. You are going to have an AFM cantilever with a molecule attached to it, to the tip of the cantilever, and at the glass surface you're going to have another molecule of interest. And as you go down with the cantilever, you're going to interact these two molecules, and then as you pull up, you can measure what are the forces that you're going to feel on that system, on that complex. For instance, here you can have like a molecule 1, a molecule 2 interacting. As you pull apart, you can see what is the force needed to break the rupture at different velocities that you're doing it, or holding it as constant force, how long it takes for it to unbind, and things like that. At the end of the day, what you get is a force extension curve, and this force extension curve then you can use to understand what is happening in the system. If you have some idea, you might go back to the bench, do some mutations in the protein, come back, measure again, and then you can have some idea about, confirm the ideas that you had about the system. Also in the 1990s, when Gaub started that, two parallel work, in separate, came up with the idea of doing that with molecular dynamics, and they created steered molecular dynamics that you probably heard about, where you actually apply you have a spring that is holding the system on a surface, let's say, like the glass surface before, and another spring that you're pulling the system from the other side. At the time, most of the simulations were done holding the center of mass of the protein and pulling the center of mass of the other protein, and you could measure that. But in principle, you can hold one atom on one side, pull the other atom on the other side. And this was developed in 1996, 1997 by Grub Miller and also independently by Klaus Schulten. At the end, again, you have a force extension curve that is somewhat similar to the one you get in the experiments, but with an advantage that you have also the position of every atom during the simulation, so you can kind of infer what is happening while you're having those peaks. It's not necessarily just the peaks, then trying to figure out what is happening. What my lab is doing, and what I have been doing even before having my own lab, is we are trying to understand how we can do the single molecule force spectroscopy in silico. Not just do one pooling, not just do 10 poolings, but actually trying to do thousands of replicas and try to get the statistics that you get from the experiments, get the same thing in the computation, and actually being able to infer what are exactly the amino acids that are making the best contributions to make something stronger or weaker. We do that by having pooling and anchoring points that mimic the experiment as the best as we can. We do, like I mentioned, hundreds to thousands of simulation replicas. We get dozens to hundreds of microseconds of our atom in B in those systems. We do dozens of terabytes of trajectory data. Just some of the projects we get to 100, 150 terabytes of trajectories to analyze. Then, to analyze that, we use dynamical network analysis. That is something that we have expanded in the last few years. And then we use dimensionality reduction tools to filter what are the most important things. the most important contributions. And we got to the point now that we're also using some AI tools for what mutations we can do to improve the system and also predict the rupture forces without simulating all of this. At the end of the day, our bread and butter is molecular dynamics simulation, right? So typically our systems have about 200,000 atoms. I don't have to explain what MD is for you guys, but I'm also part of the team that develops NAMD and VMD, QuickMD, you probably heard about. And we have this NIH center that we developed the software, and we have about 300,000 users on the software. So what we do then is try to develop things to make our life easier in solving this mechanobiology problem, at least in my lab. Of course, all the software is freely available to everyone, and we make that a big part of the lab as well. At least that's what NIH pays for. Okay, some of the tools that we developed, just because some people are asking me about these things, just earlier this year, in June, we released a new version of NAMD, NAMD 3. We have been having NAMD 3 for about 2 years in the alpha and beta versions, but now it's kind of fully operational. That's a GPU version, GPU resident version of NAMD. NAMD was the first code actually to implement GPU in the early 2000s, like mid-2000s. But the GPU resident, basically what it means, like you send all your calculations to the GPU, and there's nothing done in the CPU other than I/O. Everything is done in the GPU. We have support for multi-GPU and all the full capabilities of NAMD 2 in the same code. It's just way much faster. You're gonna see some numbers while I show my results. By December, we expect to release an alpha version of VMD2, which has been the main project in the last year and a half. And this is not exactly the new interface, but it's like kind of going that direction, trying to merge all the tools into one direction. We have changed like the surface representation, which is about 10 times faster now. We have a new secondary structure with live rendering that you can do actually up to 1 billion of atoms in secondary structures. We have implemented new ways of doing QM visualization to go faster. So all of this is going to be in VMD2 by the end of this year. Okay, back to mechanobiology. So we are interested in mechanobiology because proteins feel and react to forces in many different aspects, like, of their life. So like in eukaryotes, you're going to have a bunch of things happening. In bacteria, a bunch of different things, some of the same things, but a lot of different things as well. And you're going to have forces being important also in pathogenesis, like bacterial infection, like virus infection. And our lab is interested in understanding all these systems. When you talk about forces, one thing that you have to understand is most proteins, you're going to see here on the left, most proteins are actually producing or reacting or unfolding or doing whatever they do with a very small amount of force. Like just a few piconewtons, that's the force that they generate in the motor, or that's the force they're going to unfold, or that's the force they're going to need to unbind. Some are stronger, like strep-avidin biotin is a famous one that is like super strong. And that can hold like a few hundreds of piconewtons, maybe 200 piconewtons, 300, depending on how you tether it. And then on the other end there, in red, you have covalent bonds. What we have done in the last 10 years, it was actually 10 years ago that we published the first paper on this, we have discovered a bunch of proteins in the middle. We have discovered like that cellulosomal proteins are much stronger than strep-avidin biotin. And more recently, we discovered that staph adhesins are really, really strong to the level that they are as strong as a covalent bond without making a covalent bond. Okay, so just to give you some glimpse of like what our lab had worked with and how we work with a bunch of different people like doing the experiments. Some of this work actually started from us, some of this work started from them, some of this work our contribution is small, some of this work our contribution is basically most of the work done there. But we have studied like catch bond mechanisms in ultra-stable cellulosomal complex. That was the first one that we did with Gaub and Michael Nash, who at the time was a group leader in Gaub's lab. Now he's at the University of Basel and also at the ETH in Basel. We have studied like coronavirus evolution, like how SARS-CoV-1 evolved to SARS-CoV-2 and all the mutant variants. We are studying some of the fusion peptides, as a paper was accepted like last week with David Austin's. We have studied like filamine proteins inside, like how some mutations affect the mechanostability. Like, for instance, if you apply more force or less force, they're going to have completely different phenotypes out of these proteins because of these mutations. We have looked into like virus, like cofactors, how they influence the mechanostability of binding. We have looked at like mutations in tidine, and and so on. Like we also have, this is a paper was accepted this morning that we are studying how mechanical anisotropy is actually, like how if you pull in different places, you actually get completely different results of the forces. And we got similar results a few years ago with strep-avidin biotin. So as you can see, there's a bunch of different things happening, a bunch of different systems that are very interesting to study. They behave in different ways depending on how you apply the force. But it's still the one that is like dear to my heart is the adhesins, because they're so different. You know, like how can you make something that is as strong as a covalent bond without a covalent bond? And so the idea behind is the following: staph bacteria, they're pretty nasty, right? So they come to any surface. Typically, they're most common in binding to like a biotic surface when you have an implant, for instance, like a hip implant. They come to the surface, they start to interact with like anything that is there, including mostly of anything that is in your extracellular matrix. When they are there, there might be some hydrodynamic flow depending on where they are. This flow can be large, this flow can be not so large. Then they're going to bind, and they're going to hold into that until they start forming these biofilms. They're going to start replicating till they form biofilms. Okay? So this adhesion process should be like strong, and is a very important process in the formation of the biofilms, in formation of bacterial infection. To do this adhesion, they use what we call the M-scrams, this large matrix of molecules that are in their surface. These things can bind to absolutely anything you have. You know, like each one of these bind to different parts of your extracellular matrix. They can bind to FG beta, keratin, collagen, elastin. Like they bind to basically everything, depending on the one that you're looking at. They're all very similar in structure, and the one that we're going to show to you today, we have studied a few others. We're going to show to you the serine aspartate repeat containing proteins. Particularly, we're going to show the SDRG that binds to FG beta, and SDRD that binds to the SG1 in our skin. Okay, so how do they look like? These adhesins look somewhat like this. You have the bacteria on this side, and you're going to have the human on the other side. So the orange part there is human. The rest, like the blue and the green, is all part of the bacteria. And at the end, you have these B domains that works as shock absorbers. We showed that a few years ago. And you have this A domain that is the one that binds to the bacteria extracellular matrix. Okay? So the way they bind is they form this strand that they call the locking strand that goes on top of the FG beta, of the target protein. And then by doing that, they have this latch on, where is it? Here you go. We have the latch on one end, and then this thing closes on top and forcing this thing to be like this human protein to be kind of in this groove with a latch on top. When they first came out with the structure of this system, they thought that there are a few phenylalanines in this area were actually responsible for this to be like a strong bond, which we have shown that is not true. But the whole idea is like they came up with a mechanism that they call the dock, lock, and latch. And the idea is that you're going to have the human protein coming in the middle, that's the dock. The locking strand will come on top and lock, and then the latch is going to close and form this whole complementarity of this green part here that I'm showing that goes through these two domains, the N2 and N3 domains of the adhesin. Okay? So when we pull on that, the experiments we did was we did some SMD where we hold on one end, pull on the other end so we hold on one end of the bacteria protein, pull on the human protein on the other side, and the same thing we done the AFM to compare. At this time, I was visiting Munich, and so Lucas, there's a student from Hermann Gaub, and I were working on this, like thinking about what systems we could study that would be also strong, like we were studying the cellulosomes before. And so Lucas started doing the work with this system, and I started simulating the same system. And then when Lucas got to the lab and did his first experiment, he came back up in the room that I was, and I think I broke the microscope, because it was so much stronger than anything that they have ever seen. Because the peaks were the fingerprints that they have are these tiny things. Typically, the fingerprints are just like half of the size of the main peak, and this is crazily different. Lucas then kept doing a lot of this, trying to do different things and seeing that he was getting the same result every time. And I started downloading my first results, were actually showing that the system was also crazy strong. And this was actually we were like, we are into something. There's something here that is different than what we have seen before. We did that, like thousands of replicas of that simulation. We tested like a bunch of different mutations also in the experiments, trying to see if we could understand the system better. Just to give you some idea, whoa, it's not mine. To give you some idea, this is like the recent version of that plot. So these are like thousands of replicas of simulation that we have. This is also because that was not published in the original paper, but we have also some coarse graining where we're trying to get all the way to the speed that you do the experiment. So these are the results from the experiments that I'm excluding from the fit, and these are the results from the simulations that I'm fitting using this Dudko-Hamazabu model, and I'm getting like a pretty decent result. The result is actually better if you ignore the coarse graining, but the coarse grain actually gets closer, so that's why we like to show it. And we can see that we can actually reproduce all the results from the experiment, but also we can also look what is happening in the side of like what atoms are doing while that happens. To understand these differences here better, now we are working on something else with Tom Perkins, like Tom, like kind of going to what Paul was talking yesterday. When we got the first measurements, the peak from Herman Galb's lab was pretty clean, while our peaks always had these extra peaks right after. And then Tom Perkins, a few years after, I think this was published in 2018. Well, 2020, Tom contacted me, like he was trying to do the same system, and he was getting these extra peaks. And like kind of like we were saying, like he actually started to measure the same results. And that's because Tom has a different AFM, like with a different cantilever that has a much better resolution, so he can start to see that because now he has the resolution to see it. But we are also interested in understanding with him why the DHS fit does not work so well depending on the constant of the spring that you're using to pull the system, and like what is the variance on that, because the DHS model does not include that. But okay, so why is the system so strong? It actually is a very interesting, simple mechanism that it forms a bunch of hydrogen bonds, okay? And these are all back-bone interactions, and all these bonds actually are twisted. So because they are twisted, what happens is you have flexible linkers on both ends. As you pull on the system, like as you apply force on the system, you always have bonds in every direction, so you cannot peel it. Because as you start peeling it, you actually align. When you align, you always have the perfect shear geometry. By having the perfect shear geometry, you form all those hydrogen bonds, which vary between like 13 and 17, you know, like depending on how you count them in the MD. And that's apparently is enough to make this as strong as a covalent bond. Okay, so there is a number of hydrogen bonds that are going to make this as strong as a covalent bond. And that was a very interesting result also because it shows that only back bonds are important. And we made a few tests and got those results. Like we could confirm that with experiments. But there was still a few questions that we had. For instance, is this a hyperstable catch bond? And if you don't know what a catch bond is a catch bond is a bond that if you apply force on it, it lives longer than if you don't apply a force on it. So imagine, like, if you apply a force to break anything, it's going to live less, right? Like, you're going to break it more easily than if you don't apply a force. These are typical slip bonds. The catch bonds is if you apply a force on them, they actually resist more time. They actually hold more together than they would hold before. Somehow they look like a molecular finger trap, you know, like as you apply force, you actually get more and more locked. Okay, so to check on that, we actually had some data that came out of Yang Zheng's lab in Singapore, and they were getting some ideas that if you're holding that protein on a magnetic tweezer, and the reason why we could not do that before is because the AFM cannot hold the protein for so long, okay, like this. In the magnetic tweezer, they were showing that they could hold the protein, they could hold under force the complex for 7 hours without rupturing. And the AFM has too much drift, you cannot actually see that. We did some simulations and we were able to show that if you apply, the kind of mimic the magnetic tweezer experiment versus the AFM experiment, you actually show that if you apply, the more force you apply on the system, the higher it is the correlation of motion between the latch and the main part of the protein. Meaning like you're actually making the protein more rigid as you apply force, making the complex more stable. And that was quite surprising, and that's something that we published last year. And the idea was actually we could show that this has like two different pathways of dissociation. You have the bound state and you have the unbound state. But to go between the two, if you are under low force, if you are under no force, you're going to open that latch, open the lock, and the peptide is going to leave. Well, if you do that under high force, that cannot happen. And you have to kind of slide through the pocket, and that you have to break 17 hydrogen bonds at once. It makes it very hard. So you actually block the low force dissociation pathway, and you only allow the high force dissociation pathway to happen. And that's why this system becomes so strong. That's interesting because this is the third cash bond that we actually discovered a mechanism, and that's a very different one than the ones we've seen before that were mostly like changes in geometry that would make them more stable. This was actually only like the correlation of motion of the system gets stronger as you apply force on it. Okay, so sounds all interesting, right? But like, how does this affect the way these adhesins interact with our cells, or the way the bacteria interact with our cells? And this is the most recent work we have in the lab. I mean, the most recent one that we submitted for publication, that we start working with Yves Dufresne in Belgium. His lab does a lot of work on these bacterial adhesins, and we were interested in more on the work going towards the cell level. So we wanted to see how much we could predict on how these bacteria interact with cells. The way he did his experiments is he has SDRD, one of these adhesins, expressed on top of Adelactis, this bacteria, and then he has a glass surface with some corneocytes, so like skin cells, and then he would come down with his AFM, interact with those skin cells, and come back up. And he would do that with the bacteria with the SDRDs and bacteria without it. Okay, so the SDRD, again, from staph, not from lactis. So when he does that, if you see here the positive cases, as you go down, this is just the topography of the cell, and this is the adhesion profile. You're going to see all these white spots here, and if you look at the forces there, like 4 nanonewtons. So that's how much the force he would get, how much the rupture force would be when you go down in that area. And you'd see all in these corner areas, like around the bacteria, around the human cells, you would see this interaction. If you do that without SDRD, it does not bind. So there's nothing. So SDRD is the one that is making the connection to the human skin. That's pretty cool. We could just get some measurements, like we got the adhesion profile, the adhesion frequency we got is like 70-something% against almost 0 without the SDRD. And so with that, we could conclude the SDRD mediates the interaction between Staph aureus and the corneocytes. Then we went to the next step. Okay, what is the target in the corneocytes? Because corneocytes are cells, right? So what is the target? We then start looking into the SG1, and that's because it was already proposed that this desmoglein 1 is actually a good target for this bacteria. And so we start looking into that. These are like cadherin repeats, and we actually showed that they could bind, and bind pretty well to SDRD, so it seems like now we're doing single molecule for spectroscopy again. It looked like we are correct on the SG1. But then where in the SG1? Because you have this model that you don't know exactly if it's going to bind in different areas. We used some bioinformatics analysis, and we got 2 different possible connections. One was what we call this distal peptide, the one we call the proximal peptide, closer to close to the membrane of the human cell. Okay, so let's look into that. So like Priscilla in my lab, she started doing a lot of simulations and trying to understand the structure of the system. So the distal one would basically look like the SDRG case we had before, while the proximal one would have actually a cadherin domain kind of like smashed against the adhesin. Okay, so again, we went ahead and we started doing molecular dynamics with it. So we look at some simple metrics like mmGBSA for energy, and we could see that the proximal peptide was actually better than the distal one, but of course that measurement's not that not so reliable. We then did the pooling, and we got that the rupture force for the distal one was actually quite low, like in the just over a nanonewton in the simulations, versus more than 3,000 on the proximal one. So that looks like the proximal one is our target. But still, you want to confirm that. Then to do that, we looked into the experimental profile when you're rupturing this system. And when you're pulling it, you actually see the sawtooth shape. And the sawtooth shape is because you're actually unfolding the B domains before you actually break the complex. And sometimes you unfold 3 of them, some find you unfold 2, some find you unfold 1, some find you're going to unfold all the 5. So when we do that in the simulations, what we see is that if you get peptide 1, you should actually see the peaks that are corresponding to the cadherines, because they're going to unfold before the B domains. And when we did that with the proximal one, we actually got only the profiles of unfolding the B domains and not the cadherines, because those are actually, and that looks much more similar to what you got here. It's just upside down, okay? It's basically the same profile that you had. So the proximal peptide seems to be the main target for this system. So again, we did a bunch of replicas. This is the simulation, this is the experimental results. You can see here, what you're seeing is this shaded color there is because we actually saw a second mode of binding that I can expand on that later. But this is the result from the simulations. We also got a bunch of plots. And here I can give you some ideas about how much of that is actually done. So the system is about 200,000 atoms. The final results that we used to analyze and submit in the paper, it was 107 microseconds of MD per atom, 760 microseconds of coarse-grain MD that we did with GROMACS. This was like our atom was done with NAMD3 and Charm36. To give you some idea about the speed of doing that in the new version of NAMD that I mentioned before, our best results so far is with the new Delta AI machines, which are these Grace Hopper 200 nodes, and we got 381 nanoseconds per day per GPU. So that's like for a system of 200,000 atoms, which I think is pretty decent, like we're expanding a lot on that. So with all those results, what do we see? One of the interesting things that we noticed compared to the SDRG that we had a lot of experience is that this system forms this pi stacking around the region where the lock strand is, which calls attention because it was not there in the SDRG. And, but mainly, when we did this correlation analysis using our dynamical network analysis, what we notice is you have regions here that are actually connecting the protein, the adhesin, to the cadherin. And this is the area that you actually would see the normal latch interacting with the binding peptide, the human peptide. But the cadherin is a new thing. We didn't have that in the SDRG because that was actually the terminal part. So that was actually pretty interesting that there's this extra component of the cadherin that could make this system act a little bit different than the other ones. Well, okay, if the cadherins and also these B domains, another thing that calls your attention is like how does calcium influence on that? Because cadherins are full of calcium, B domains are full of calcium. And then we actually, and we knew actually from some previous results that calcium is important for the B domains. When we looked into the system, like the regular pattern for the system when we were doing the first experiment, it looked like this, the pooling plots. When we add DTA, you're gonna kill all the B domains because they depend on calcium to be like folded strongly. So you remove calcium, and then you get just the peak of the adhesin, just the peak of the binding, because that one does not depend on calcium. Then when you add a lot of calcium to it, you actually always get 5 peaks. So you always get all the 5 B domains unfolding. But more interesting than that, we actually get that the adhesin is stronger. So what the forces we had before were like just over 2,000 piconewtons, like 2 nanonewtons. And now we're getting like 2.6, 2.5 piconewtons to nanonewtons as the force of that system, which makes this system the strongest complex ever found, stronger than the SDRG we had before. But okay, why is calcium influencing that? The first idea we had is like, okay, because we have all these sharp peaks of the B domains, and B domains are shock absorbers, maybe the B domains are influencing the adhesin. But actually, we showed the simulations that that's not true. You know, like removing B domains did not affect the A domain. But we actually decided to test something that our colleagues could not believe us when we told them that if you actually look at the cadherin domain that is interacting now with the adhesin domain, the cadherin domain is going to have this calcium there. If you remove that calcium from there, so like when we're pulling on the system, if we remove the calcium from the cadherin, we actually get a drop in force about 30%. So like we just remove those calciums, and then you see this drop in force because the cadherin is less stable now. Cadherins are not that strong, but remember, you're trying to unfold the cadherin is meshed against another structure. So it's actually very hard to make it like change conformation to pull it out of this tiny hole, right? You're trying to pass it through a hole. And that's actually make it very strong because the calcium is stabilizing that bond. Now you might think like, okay, what does calcium has to do with infection then? And I think that was the greatest last step of this work that our collaborators in Birmingham, England, not the one in Alabama, did this experiment that gave us cells, actually. The experiments were done by Dufresne, cells with atopic dermatitis. So now you're looking into not healthy cells, but cells that have some kind of like skin condition. And when we look into that, we always see these 5 peaks. We always see the 5 peaks because atopic dermatitis cells have a higher concentration of calcium. And then we see the adhesion profile being kind of the same in terms of force, but we see that now, instead of binding, remember, it was just a corner binding before, it binds everywhere. So it's actually much easier for the bacteria to adhere to your cells when you have your cells being damaged than it is when your cells are fine. That's why you might have a hair follicle inflamed that becomes a great place for an infection in your skin, or any scratch that becomes a great place because of conditions like this. You actually have a higher concentration of calcium. Desmoglin, the SG1, actually moves around. It's not just on the corners because of the changes in the cell, and then the bacteria can adhere everywhere. Okay, so last, like I know my time is over, so last okay, that's pretty cool, and that's like I think that's one of the greatest discoveries we did there, but we were also interested in the evolution of this system. And Priscilla, a few years ago, she noticed that she was actually using AlphaFold to create all possible structures of adhesions that we could, and trying to measure the forces. Each one of these points here is actually one measurement of force of pulling. And she started noticing that depending if they were methicillin resistant, so the ones that are resistant to antibiotics versus the ones that are susceptible to antibiotics, she was getting different results. So like there's something in the evolution there. And that's actually another story that I will let you tell you later, but we are actually doing the experiments. We sequenced all of a bunch of bacteria. We got bacteria from the local hospital. We got bacteria from the 1900s, from bacteria banks. And we're actually sequencing all of them and studying getting these models for force resilience, and we're actually seeing that they're actually evolving to become stronger, something that a few weeks ago was confirmed by my collaborator, Michael Nash, using a spinning disk technology, not AFM. With that, I just want to say that I was giving you an introduction of what we call it is in silico single molecule force spectroscopy. I hope you will see that many biological systems behave in a surprising and kind of unexpected way. This advancement of the software also, like in the exascale computing, is allowing us to now reach the same level of like sampling that you have in the experiments, which is kind of giving us new ideas about how the system behaves. Then on the bacteria side, they exploit this hyperstable non-covalent bond to adhere to us much more strongly, and calcium plays a big role on that, and this methodology that we're using can actually give us some insights about how that happens. To conclude, I just want to say that if you're interested in learning more about doing computational biophysics, we host these workshops that was created by Klaus Schulten in the early 2000s. We are going for the number 62 later this year. That's the 3rd time we do that in Auburn. And if you're interested, please join us. We have a bunch of people there teaching about NAMD, VMD, and computational biophysics in general. Also, if you know someone or if you are interested, we have a lot of positions for PhD there. Auburn is located like an hour and a half from Atlanta. We're also about 2.5 hours from some of the most beautiful shores in the US. It's not a bad place to be, other than the hurricanes are hitting us all the time now. And I think it's a pretty nice place if you want to come and visit, and also if you want to come and join us for a PhD. And with that, thank my funding sources, especially NSF and NIH for the great support. Was really nice to get big NSF support immediately after I started. And all the people in my lab, you see a bunch of Portuguese names here, there's a bunch of Brazilians there, and thank you all for your attention.
Thank you, Rafael, for your very nice work. Beautiful, really. Maybe later on you can send it to me, and I can advertise in the SBBF news. Yeah, if you have open position, I'm sure we have students that would like to go there.
Oh, sure, yeah, we have positions both in my lab and every single lab there. we are looking forward to have more people. And
Yeah, and then I can advertise it through the SBBF meeting.
yeah, we have 4 Brazilian students in the department now, and they are all doing really well, so the people are very excited about Brazilian students there.
Yes. Questions?
So when you have all this collection of hydrogen bonds, kinetically, don't you get trapped before you form the wrong hydrogen bonds and you cannot form the right ones?
Yes, you do.
And that's not a problem?
It's not, because as you start applying force on it, you kind of force the system to align again. So imagine that you have the kind of a wobbly
But
area.
if you form the wrong ones, you have to backtrack, don't you?
If you have the one with the locking strand on top, and that's the affinity there is very important. So the backbone interaction is important for the mechanical stability, but the side chains are important for the initial binding. So as you bind in the right place,
But how do you bind the right? I believe you can form the wrong hydrogen bonds with the backbone. That's what I'm curious about.
But you need side chain bonds to form this initial binding at this location under the locking strand. So you actually form those before, and then the locking strand is kind of wobbly. It's not forming all the time.
So
if you
the
look at
system
crystal
gets in the right place before the hydrogen bonds get formed. Is that correct?
Yes, and if you look at the crystal structure, you actually only have the region under the locking strand. All the rest is actually, we had to extend, like by just placing the amino acids anywhere. Because as you start pulling on them, they kind of lay on top of the rest and they form the perfect bonds.
So when you look at the simulation, do you see all these hydrogen bonds form at the same time?
They kind of, it's like a reverse peel. They come slowly and form them because of this shear geometry. If you pull, we pull like in the completely opposite direction. Instead of pulling like this, we pull it like this. Like the system flips and then it's formed the bonds again. Was quite interesting, that mechanism.
More questions?
Hi, Rafael. Great talk. So I was wondering about, so if you have this this adhesion that has this bond that is able to withstand a force greater than a covalent bond, you said that, right?
It's about the same.
It's about the same, right? So I kept
Remember
thinking
that
like
covalent bonds are like, so when I say greater than covalent bonds, like gold-thiol is the bond that we know that's the weakest one. It's about 1.8 nanonewtons. But like there are covalent bonds that resist. We don't know the number.
okay. Yeah, I was thinking, well, if I mean, because there's places in this complex where there's
they
single
would
chain,
rupture the
right?
covalent bond,
So you would rupture that.
No,
And
no,
so I
you
was
don't,
thinking-
because
yeah.
those are much stronger, actually.
Okay.
We were actually afraid of that as soon as we got the first data. We knew it was not the case because otherwise the cantilever would die, right? The cantilever has a limited number in the experiment of adhesins bound to it, like maybe like 10, 20. As you go up and down thousands of times and you're still binding, getting the same result, that means that you're not breaking those covalent bonds, you're actually breaking like temporary bonds that would form and unform.
Okay. Yeah. And then that the video that you showed of this this process happening.
Yeah.
That was a reversal.
That was a reversal.
Okay. Yeah. So
No.
that's really cool that you were able to get this process.
No, yeah, that was just a reversal. Like we know kind of how it works, so we pull it in the right direction so that we would see the binding. And although the binding is pretty complex, I talked to Chris Chipo, who works on those kind of things, and he was like, "Okay, we were never gonna figure that out with the computer." I didn't expect, but I asked him just in case.
Thank you.
Thank you.
Rafael, really nice. Every time I look at it, it reminds me of a project that I had it running regarding knotted proteins. So it looks like a knot, but instead of intermolecular, it seems like an intermolecular knot that is gas formed. And every time I tried to do the PMF in like a knotted protein, I always had this problem of I had to peel it like very small, the stirred ND was really a nightmare. And the strength was so strong that I thought that there was some problem with the force field. When you look at this, did you have to calibrate something in your force field? Was it straight out of the box?
Straight out of the box, not at all. So that has a lot to do with the velocity that you're pulling, right? Yep. And also ions. Monovalent ions are not a big deal. Calcium are nasty, so we don't get very good results for the B domains. If you see our B domains, they are good, but if you try to fit them back to the experimental results, we don't get the same results because the calcium ions are typically considered sticky in the simulations, right? Although for us they're not sticky enough. You know, like they should be a little bit more sticky. You know, like, and that's something that we are actually. I have a student right now trying to do that with QMMM, where we are trying to do the same thing but using QMMM for simulating the calcium loops. Just was more like a curiosity than a major project. You know.
Another question?
It's really very impressive how far you've been able to push these simulations on these experiments, really bringing it together and getting almost on the same timescales. I have a question regarding the catch bonds, which are a really fascinating phenomenon. So I guess on an abstract sense, if you think about it in terms of an energy landscape, the common denominator is always that it's sort of pushing it towards the barrier, so it cannot sort of go around and take the lower energy path. I was wondering, at the level of the molecular mechanism, is there also a simple common denominator, or are there very different mechanisms how this is achieved, since you've looked at several of these now? So
all the three systems that we looked at were catch bonds, we got different results. So like take strep-avidin biotin, they are super strong because you try to break the same kind of, this is kind of similar to this one, you try to break all the bonds at the same time. If you pull a different tether, you actually peel the side of the strep-avidin and then it becomes a little bit weaker. Cellulosomes were very interesting because they were kind of flat surfaces interacting, like these cohesin dockerines. And as you apply force on them, they rearrange their side chains and they form like, they increase their contact area. And when we published that, Viola Vogel, like your colleague there, was very interested in that because she could not imagine like that, like a side chain like change would actually give you like such a difference in energy. And more interesting, there was no extra hydrogen bonds, no extra anything that like is a regular chemical descriptor, you know, like that people typically look at. It was just like contact area going up. Now that we're doing more and more with this network analysis, the thing that we're seeing that is common is that the correlation of motion between the atoms actually increases. So as you pull, in all of these cases, correlation of motion between the two sides goes up instead of going down as you try to break them. I think maybe that's the only thing in common that we're having. And that's why we're trying to train AI tools now on predicting rupture forces by using just the correlation of motion.
Great, thanks.
Okay. Any other question or comment? Then thank you again, Rafael.
Thank you.
Very nice. And now we call Pedro Sodré from Federal University of ABC. It's you, Pedro? It's not you?
I don't have a talk.
Sure.
Are you sure? Eu recebi e-mail e disseram que eu não fui selecionado pro shortão. Mas eu tinha pedido.
Então, ele acabou.
Tudo bem.
Não, mas aí não é. É que você não recebeu. Desculpa. Peraí.
Sorry, it was my mistake.
Hello.
Sorry. And then I'm going to introduce Dr. Felipe Cardoso Ramos from Unicamp. And he's going to talk about computational investigation of tryptophan fluorescence in DPO4 mutants through classical approach. You have 12 minutes.
Okay, thank you for the introduction. So good morning.